Welcome to Bioratna. Dear students, since last week we have started the topics on genetics from standard 12. Since your class 9th, you have come across many terms, the concept of which might be clear to you or not. Also, there are many terms which are not in the book, but it is so obvious that you have to know about them to have a complete confidence in your subject. Similarly, there are many terms very frequently used in the topics which the students find difficult. So every Sunday, rather than having a different topic, we will be having a special video in which all your concepts will be cleared based on whatever topics have been taught in the previous week. We might add other things also, like latest development in biology, latest researches, latest developments in biology, which will keep you updated. So let us start with the first one, that is the difference between gene, DNA and chromosome. You know what is gene, what is chromosome and what is DNA. But when you are asked what is the difference between DNA and chromosome, let us see. Chromosome. Chromosomes are the hereditary material present in our cells. If it is eukaryotic cell, it is present inside the nucleus. And if it is prokaryotic, then naturally it is in the cytoplasm. It consists of DNA as well as protein. The DNA, which is the genetic material, it needs the help of some proteins so that they are packaged properly inside the nucleus or in the cell. So chromosome consists of DNA as well as protein. Now come to DNA. That means it is a part of chromosome. So DNA is actually the genetic material, which is a polynucleotide, And what are genes? Genes are the segment of DNA also you can say, or chromosome also you can say. So genes are the part of DNA which controls the formation of, you will say, proteins, that is enzymes. Yes, of course, they control the formation of proteins, which are enzymes, as well as they help in formation of RNA. Like tRNA, rRNA, they do not further produce any proteins. But if they are producing mRNA, then it will express itself in the form of protein. So I hope your concept about what is gene, DNA and chromosome is clear. Let us move to the next one. Karyotype. What is a karyotype? What is a karyotype? Karyotype, it is the complete set of an individual's chromosomes which are arranged as per many criteria are taken into consideration, like they are arranged as per their length, as per the position of centromere. What is centromere? You remember, you have studied in class 11th. Centromere, also called the primary constriction. So there are many other factors also. Accordingly, they are arranged and then they are numbered also. For example, in case of human beings, there are 23 pairs of chromosome. Means after arranging the chromosomes, then they are numbered. Numbered like number 1, 2, 3, 4 like that. And this roll like the roll numbers which you have. But these roll numbers cannot be changed. This is chromosome number 1 means this is chromosome number 1. So this is karyotyping. What is it used for? Karyotyping study helps in many things like it helps in the study of chromosome aberrations. It helps in the study of evolutionary history. Few questions asked on karyotyping is like you are given the photograph of the karyotype and then so this is chromosome number 21. We are having 23 pairs of chromosomes. That means in the karyotype, what should be the number of chromosome 21? 2. But in place of 2, you will be shown 3 chromosomes. 1, 2 and 3. And you have to identify by studying the karyotype which disease it is. Which disease it is. So this is karyotype. Now let us move to the next one. Next term is haploid and diploid. This is what is haploid and what is diploid. You know that human beings, they are sexually reproducing organisms who are having 2 sets of chromosomes. The zygote, which is formed by the fertilization of the 2 gametes, each one is carrying 1 set of chromosome. 1 set means what? Like you are studying 5 subjects, physics, chemistry, bio, English and 1 optional one. So if you are having books for all the 5 subjects, that means you are having 1 complete set. That first set of course is NCERT and the second set may be any other if you are having that. So if you are having, when the both sets are present, is called diploid and which is present in somatic cells. This diploid number, it is present in all somatic cells. But during the formation of gametes, the number is halved. So that the gametes, they have only 1 set of chromosomes. This is called haploid. This is haploid set will be present in the gametes. This we are talking about human beings. This is not the case about everyone. This is also true about flowering plants. This is not true about all. This we are speaking about human beings. Clear your concepts with the help of this example first. So diploid means having 2 sets of chromosomes, and when you talk about haploid, 1 set, when the 2 sets are separating, then it is not so कि एक set में 2 books of biology you can put, in another one you can put 2 of physics, not like that. 1 each from each subject, then only it is going to make a complete set. Otherwise, it is going to give rise to some disease, some kind of abnormality. This also happens. Just now you saw Down syndrome, where there was 1 extra chromosome number 21. But that is not the normal case. So this diploid is written as 2n and this haploid is written as n. Haploid and diploid. Clear? Next, what are homologous chromosomes? What are homologous chromosomes? Without having a clear concept of homologous chromosomes, you cannot take a step ahead in genetics. What is homologous chromosomes? Just now I told you that in diploid organisms there are two sets of chromosome, one inherited from father and another set inherited from mother. So one complete paternal set will be having chromosome number 1, 2, 3 to chromosome number 23. Similarly, the maternal set, it will also have chromosome number 1, 2, 3 and then up to 23. I told you that this number cannot be changed when we arranged the chromosomes to form the karyotype. To see the karyotype, at that time they were numbered. Chromosome number 1, paternal 1, is exactly similar to the maternal chromosome number 1 in case not only just size, not only just the position of centromere, but the different genes which they have. Paternal chromosome 1 and maternal chromosome 1, they are homologous to each other. This is true for all the autosomes. This is true for all the autosomes. Only in case of sex chromosome, X and Y are not homologous to each other. X is much longer. Chromosome is having many extra genes which are not present in the Y chromosome. Understanding what is homologous chromosome will help you in understanding the term gene locus. See what is gene locus. Gene locus. Fine. Let us take any one pair of homologous chromosome. This is one chromosome we have drawn. Exactly similar will be its homologous chromosome. Suppose this is maternal and this is paternal. Then if this chromosome is having the gene for a particular character at this position, the position of a particular gene on the chromosome is called gene locus. तो उसका जो homologous chromosome है ना, उसमें भी exactly same location पे ये gene होगा. During meiosis, when exchange of material takes place, linkage and crossing over takes place, this is only possible. ऐसे ऐसे लिंक करते जाओगे, जैसे जैसे पढ़ते जाते हो ना बच्चों, वैसे वैसे देखोगे everything gets linked to something other. तो ये दोनों सेम जीन लोकस पे है. So when they come together, they can exchange their genes. If this is A1 and this is A2, then after exchange, A2 will come here and A1 will move to the other side. This causes variation. This was gene locus. Now, today's last term, the pure breeding lines. Pure breeding lines. What are pure breeding lines? What are pure breeding lines? There are many plants which undergo only self-pollination. Their stamen, pistil mature at the same time. Their corolla, it remains closed all the time. So it is undergoing self-pollination generation after generation. So there is no question of variation. No foreign gene is coming to it from other flower. So having undergone self-pollination generation after generation, those plants which show stable inheritance of characters, are called pure breeding lines. What is stable inheritance of character? If the plant is tall, it is having both alleles the same, capital T and capital T, no question of small t. So when it is having only capital T, no other allele, then every generation it will produce 100% only tall plants. That is stable inheritance, no fluctuation. So this is all for today, students. See you coming Sunday with another set of terms. If you are new to the channel, then do subscribe and hit the bell icon so that you get all the notifications in time. Thank you for watching the video.